Good afternoon. So I'd like to talk a bit about our efforts to develop newer treatments for patients living with advanced GIST and trying to move beyond imatinib, sunitinib, and all those other IBs to newer agents. There's been a lot of interest in the last decade or so in many human cancers in targeting Hsp90. And this has been of particular interest in GIST. And I'm going to describe our clinical trial with an Hsp90 inhibitor in the advanced GIST population. Trying to explain what Hsp90 does is difficult, but it is a heat shock protein, and basically what it does is it helps other proteins fold into their final shape so that they can be functional. So when a protein emerges from the ribosome, it's a string of amino acids, and that string has to be arranged into a three-dimensional shape so that the protein can be functional. This is true of many different proteins in the cell. That is also particularly true of proteins involved in the cancer phenotype, in the oncogenic signaling in GIST, and that includes proteins like KIT, both mutant and wild type, and PDGFR. So it seemed scientifically to be a sound idea to try to block Hsp90, inhibit the folding of KIT proteins, and therefore inhibit KIT signaling, and possibly find an effective new treatment for advanced GIST. In addition, this strategy should not, in theory at least, be vulnerable to mutations, the same way those mutations that Benedict showed could block the binding site for imatinib. Even a mutated KIT needs to be folded, and therefore Hsp90 inhibition could be a potential way to attack such a malignant phenotype. So in the last decade or so, many different Hsp90 inhibitors have been tested in the clinic. These are just three. It all started with geldanomycin, which is actually a naturally occurring antibiotic, which was modified to form tenespamycin, or 17-AAG, which is one of the earliest Hsp90 inhibitors in clinical trials. A lot of issues with this, mainly involving getting it into a human safely. It has to be formulated in certain substances to dissolve it and get into the bloodstream, and it's not that easy to give to patients. So another company, Infinity, developed BIIB021, which many of you may have heard of before. This was tested in advanced GIST patients in a large clinical trial that was reported about 2 years ago at ASCO, run by the Dana-Farber group. And because of their encouraging preliminary results, they designed a large phase 3 study to test about 200 patients, randomized. Half would get BIIB021 and half would get a placebo control to see if it made a difference. And unfortunately, that study was stopped early after only about 45 patients were enrolled because there are an unexpectedly large number of toxicities, including 4 deaths related to liver toxicity. This is a signal to all of us why we have to proceed so carefully and methodically and sometimes frustratingly slowly in developing new drugs, because the risks to patients are potentially real. With the end of the BIIB021 story, we've started looking for other ways to target Hsp90 in a safer and potentially effective way. BIIB021, you don't need to be a chemist to see, is very closely related to 17-AAG, the original Hsp90 in structure. But the drug I'm going to talk about today, BIIB021, which is made by Biogen, the company formerly known as Conforma, is a completely different type of molecule. It's completely synthetic, and it seems, at least structurally, to attack Hsp90 in a very different and unique way. The other interesting thing about it from a patient perspective is that it has the advantage of being formulated as a pill, so it's an oral medication rather than an intravenous one. So this is the drug that we had the opportunity to test in a phase 2 study. And as we design phase 2 studies, we first look at the safety data from the phase 1 study where the drug is given to human beings in the first place, and that gave us information about what we thought would be a safe and reasonable dose to try in patients with GIST. And then we decide how we would assess the potential efficacy of this drug. And we know from Gleevec, for example, that within days of a dose of Gleevec, a PET scan turns cold, the SUV numbers fall, and that's a good way to see that you're having a biological effect on a GIST tumor. So in this case, we used early PET scans as a way to measure the potential effect of this drug, this Hsp90 inhibitor, using also a lot of secondary objectives to look at various blood tests, pharmacokinetics, and correlative endpoints. This was a pretty standard phase 2 study, and I just want to point out that all of these patients had advanced metastatic GIST. None of them could be cured with surgery. All of them had previously been treated with imatinib, and it had progression of disease, and sunitinib, and it had progression of disease. So these are patients who are looking for their third-line treatment, if not fourth or fifth line. They had to meet standard eligibility criteria in terms of blood counts, basic serum chemistries, and so on. And also because we weren't sure whether it was safe to combine drugs. In other words, could you give this BIIB, or BIIB0 as we fondly call it, could you give BIIB0 in combination with Gleevec or other drugs? Everything had to be stopped for at least 2 weeks before you start this clinical trial drug. So this was the design of our study. And based on the phase 1 study, we had decided to give this as a rather interesting schedule, which is twice a week. So patients would take 600 milligrams of oral BIIB0 twice a week. So for example, on Monday you take your first dose, on Thursday you take your second, and on Friday you get a PET scan, and that first PET scan would give some sense of whether there was a beneficial effect, at least on the PET scan in terms of the SUV uptake of the tumors. And then this was pretty intensive for the patients. They had another PET scan on day 8. So again, you're treated on Monday, you're treated on Thursday, you get a PET scan on Friday, you have the weekend off, and on Monday you get another PET scan. And that third PET scan is done before another dose of drug is given. And the point of that is to see if the effect has worn off over that weekend when no drug is given, to see if going 3 or 4 days between doses was too long to last. And when we look at the results of this first look, the patients with the black bars are the ones who are receiving the twice-a-week schedule. And you can see that for many of them, the black bars are pointing up, which means that over that weekend break, the SUV has increased modestly from 10 to 20% for many of the patients. So that led us to be concerned that this drug was being given perhaps at the right dose, but not frequently enough. So we changed the schedule and now gave patients 400 milligrams, but 3 times a week. So the same total weekly dose, 1,200 milligrams per week, but just divided up in a slightly different way. I hope that is clear. So in total, we treated 23 patients on this study, and they were, as you'd expect, a mix of ages from 33 to 88. And all of the patients, like I said, had received prior imatinib. All of them had received prior sunitinib. About 60% of them had received prior sorafenib, and 2 of them had had nilotinib. So really, these are patients who have seen a lot of prior therapy for their GIST. And I'm going to cut to the punchline, which is the results that we saw after a month of treatment. So patients have a PET scan. If you just forget about that weekend description for a moment, patients have a PET scan before starting, and then get continuous treatment for 1 month, and then have a repeat PET scan done. So this is after 4 weeks of treatment. And we compare the average SUV as measured by the PET scan from beginning to end, 28 days later. And what you can see, first of all, is that a few patients had actual responses, and this was defined as a 25% decrease in the mean SUV max. So a few patients have managed to get the bar to go down, 50% decrease, 40% decrease, and so on. And many other patients had some degree of decrease in their SUV activity, but it didn't quite meet that arbitrary cutoff. And then other patients didn't seem to have a benefit and got worse despite that. So this, to us, was encouraging that you could at least get a signal of efficacy of affecting the biology of this tumor. This is one of the patients who met the formal criteria for partial response and had a greater than 25% decrease. And you can see the pre-treatment PET scan on the left showing extensive black spots consistent with hypermetabolic metastatic GIST involving the abdominal wall, the liver, the mesentery, the bowel. And you can see after 29 days of treatment, there was a significant improvement in at least this one patient. And of note, this is somebody who actually had to stop their imatinib or sorafenib or whatever their prior treatment was, and was on only the BIIB0 drug during this 4-week period. So overall, we found that we had 5 patients with official partial responses, and that gave us a total response rate of 22%. There's a lot of controversy in medical oncology on the best way to measure responses to new treatments for GIST. Some people do PET scans, some people do CAT scans and use the so-called response evaluation criteria for solid tumor. And some people do CAT scans and do Choi criteria, which is a combination of looking at the density on the scan in addition to the size. And again, depending on how you choose to draw a cutoff, the actual response rates vary from 0 to 22%, again, depending on which method you're using to measure. But there's at least, I think, as you can see from this PET scan, a hint of something effective going on there for patients with advanced GIST with an Hsp90 inhibitor. The other big question in any study of Hsp90 inhibition is, was the drug safe? Was there significant toxicity? Because I told you about the Infinity results, which had significant liver toxicity. And I'll just highlight down here the liver enzymes, the AST and ALT. And what you see across the board really is a whole lot of zeros. And the final point is that there's really not a whole lot of toxicity that we observed in this study for this drug in this population. And that is encouraging. However, again, this is a smaller study than the Infinity study, and clearly if we treated more patients, a safety signal that we didn't yet observe could appear. And the other thing we did is look at the drug levels in the blood. You know, people are interested in looking at imatinib levels in the serum, and for this new agent, we were also interested in seeing what kind of drug levels were being achieved in patients. And this plot looks at two things. One is what's the maximum concentration in the blood of the patient of the drug, and two, on the y-axis, what is the change in SUV max in terms of the PET scans. And again, this is a preliminary analysis, but what you can see is that many of the patients who did poorly, meaning that the SUVs went up 100% or 140%, those patients tended to have lower levels of the drug in their blood. And what you see is that patients that benefited from the treatment in the sense that their SUVs went down, those patients included some who had higher levels of drug in their blood. And although there are some data points here, there are some here, and there are some here, in this area you see not many data points at all. In fact, no patient with a high level of drug in their blood, say over 2,000, had an increase in their SUV max. So that again suggests that the drug is on the right track. Maybe we need to get higher doses of it into patients, or maybe on a different schedule, but I think it's at least a start. And to acknowledge the people who are participating in the study, this is a small list, but many more were involved. Our colleagues in pathology, Christina, of course, radiology, and the Mayo Clinic, who treated about a third of these patients. I should have mentioned that at the beginning. And in terms of Hsp90 inhibition in the future, one thing we learned is that many of these patients, although they seemed to do well for a short time, eventually had disease progression and had to go back on some other treatment like Gleevec or Sutent. And when you stop Gleevec or Sutent on a patient who's been slowly progressing but is otherwise holding their own, things can sometimes get worse rather quickly. And to force stop a drug that's holding the disease at bay to go on a clinical trial is challenging. And in the future, I think patients are going to have to continue their Gleevec no matter what, and we're going to have to find regimens to add to that to try to control the disease where the Gleevec is not enough. And that is, in fact, the next step. There's an ongoing Hsp90 study that's combining a new drug from Astex, I believe, with Gleevec. And our next study here will be a PI3 kinase inhibitor in combination with Gleevec. So that, we think, will be the way forward beyond the IBS, the imatinib, sunitinib, sorafenib drugs for the future of systemic treatment for advanced GIST. Any questions?
I just want to know if Gleevec was the drug that was working best over the years, and these drugs for people who no longer get benefit from Gleevec?
Correct. Yes,
And Gleevec is still the number one as far as treating?
Gleevec is still the #1 first-line treatment for GIST.
Yeah. I
Yeah.
guess I have another question. Martin's doctor told him that since he's taking Gleevec for, I think, 10 years or so, he said chances are those who stopped taking Gleevec because it wasn't working were those in lesser years, fewer years that they were taking. And he had the opinion that if one is taking it for 10 years or so, that it, in all probability, will continue to work. Did you find that?
Many of these patients had been on Gleevec for variable times, but I think the average time since diagnosis was something like 5 years, 6 years, 8 years. But resistance can develop at different times for each patient. But I think the longer you're on it, the better. And if there's no evidence of the disease worsening, then carry on. I mean, Hsp90, which became Gleevec, was only available in the late 90s, I think, ballpark, for the first time. So no one's been on it 20 years yet because it hasn't been around yet. But I'm looking forward to meeting that person.